You're watching Coronavirus in Context. I'm Dr. John Whyte, Chief Medical Officer at WebMD. There's a lot of talk about vaccines. Some folks are saying it's going to be available by the fall. Others are saying it's years away. To get insight into how realistic vaccine development is, I've turned to Dr. Paul Offit. He is the director of the Vaccine Education Center and professor of pediatrics at CHOP, Children's Hospital of Philadelphia. Dr. Offit, thanks for joining me.
My pleasure.
So where are we on the possibility of a vaccine? Is 12 to 18 months realistic?
Well, remember, the average length of time it takes to do the research of development on a vaccine is about 20 years. And that's
20 years, okay. All right.
I was fortunate enough to be part of a team at Children's Hospital of Philadelphia that created the strains that became the bovine human reassortant rotavirus vaccine, Rototec. That took 26 years. That's about average. If you go through all the steps, that's about how long it takes. If you're trying to get a vaccine out in 12 to 18 months, then you're going to have to skip some steps, major steps.
Okay. What are those steps that might be skipped?
So here's typically what happens. Typically what you do is you do so-called proof of concept studies. This is a virus that we just have in hand now only a few months ago. So the question is, how do you want to make the vaccine? Do you want it to be a whole killed virus like the inactivated polio vaccine or the hepatitis A vaccine? Do you want it to be a live weakened form of the virus like the measles, mumps, or rubella vaccines? Do you want it to be just a purified protein like the hepatitis B or papillomavirus vaccines? Do you want it to be a vectored vaccine, meaning a virus, another virus, which is innocuous that you then sort of clone in the gene that you care about, which is in this case the gene that codes for the surface protein of coronavirus, the so-called spike protein, and that's the way the Ebola virus vaccine is made, that's the way the dengue vaccine is made. Or do you want to try something that's never been done, which is a messenger RNA or DNA vaccine, where basically you give the gene or message that codes for that protein, the spike protein, so you don't know. So normally what you would do is you would do a proof of concept. You would have an animal model that would get sick when you inoculated it with COVID-19. Then you would try one or another strategies and see which one worked. Then what you would do is you would do dose ranging trials, trying to see what dose I can give to induce an immune response that at least in animal models correlated with protection. Then you do larger and larger trials, usually hundreds or thousands of people, to do a big dose ranging study. Then you do a phase 3 trial, the definitive FDA licensure trial, which typically is a prospective placebo-controlled trial. For our vaccine, that was a 70,000 child
70,000
4-year trial, and that's typical. The Rotarix was 60,000 person trial. Conjugate pneumococcal vaccine was a 30,000 person trial. The HPV vaccine was a 35,000 person trial, roughly.
So why are we talking about 12 to 18 months? Is it, some people say, well, we had data on SARS and it's similar. Others will say that the UK is pretty far along. And we're really waiting on many of our mitigation strategies with the hopes of a vaccine might be here soon.
So what would you skip? I think you skip the animal model studies. You do small dose ranging studies, which Moderna, which is making an mRNA vaccine, has already done, or Inovio, which is making a DNA vaccine, is in the midst of doing. And then you say, okay, here's a dose that induces an immune response similar to that occurs after natural infection. I'm going to assume that that immune response is protective, even though I don't necessarily know that.
Right.
Then what you would do is you wouldn't go and get FDA licensure. You wouldn't. You would do a trial, a prospective placebo-controlled trial of 1,000 or 3,000 or 6,000 people, which may or may not be enough to tell you whether it's effective. It's certainly not enough to tell you whether or not you have any sort of uncommon side effect problem. And then you would roll it out. This is basically the Ebola vaccine model. That vaccine was rolled out into West Africa without FDA licensure. I mean, and so then when you gave it to tens of thousands of people, you could see how effective it was. You could see how safe it was. And then you got a license. That's the way that worked.
And how long did that take? How long did that take?
How long did it take to do that? It still took a couple of years to do that.
And was it effective? How effective was it?
It was effective. It was effective. It had one mild safety problem, but it was an effective vaccine.
Well, safety is always an issue, as you know, particularly on vaccines. So we take all of this into consideration, and there are current, phase 1 trials currently underway for various vaccines. What about the fact that folks are saying, you know what, we can look at the data as the most that we have, and we can make some decisions in a few months about manufacturing, because otherwise, then we're pretty far out. What are your thoughts on that?
I think what they would do is manufacture at risk. In other words, they would manufacture, scale up without knowing whether or not the product that they're making is safe and effective, in the hopes that it's safe and effective. And the government can mitigate some of the risk there. BARDA has had, which is
Hmm.
part of Health and Human
Hmm.
Services, has given $500 million to each of 5 different companies to
Sure.
mitigate some of that risk.
But again, what are your best estimates? Because in many ways we say if all the stars align, which they don't usually, right? It usually is trial and error. We often don't get things right the first time. Do you have a sense of timelines of how you think it'll play out?
It depends on what you're willing to accept. I mean, if you're willing to accept that when this vaccine first is introduced into thousands or maybe even tens of thousands of healthcare workers, frontline responders, grocery store workers, pharmacy workers, mass transit workers, so I think that's who this would be tried in first.
Mm-hmm.
If you're willing to accept that when that vaccine is being given, you haven't done a large safety and efficacy trial. So to some level, it may not be as effective as you think, or it may not be as safe as you think, but you're scared of the virus. I mean, you're still
Mm-hmm.
panicked about the virus because it's still killing 1,000 or 2,000 people every day. Then you're willing to accept some level of risk. If at the time the vaccine is introduced, it's killing, say, 10 people a day, you may not be willing to accept that risk. And so I think it's all risk benefit in medicine, right? I mean, we give cancer
Right.
chemotherapies that can kill people to people who have cancer because you're willing to take that risk because the cancer could kill you.
But we're often not having that narrative in the media. We're just talking about a vaccine being available. And you're right, there will be caveats to that in a discussion of risk versus benefit. And sometimes people misestimate the risks of vaccines.
Right. So I think you at some level have to manage expectations. I think that if you haven't given this vaccine yet to tens of thousands of people, so you can't say with confidence that how effective it is, or you can't say with confidence that it doesn't have an uncommon side effect, then you have to manage that expectation. The other expectation you have to manage is how well this vaccine will work. I mean, I think it's a coronavirus, so it's going to be like other coronaviruses where the protection, even following natural infection, is usually short-lived and incomplete, meaning it's not going to last for decades. It may last for a couple years, and it's not going to protect you against asymptomatic reinfection or even mildly symptomatic reinfection. You're trying to just keep people out of the intensive care unit and out of the
Right.
morgue.
Would we give it to children, or do we need to study them separately?
I certainly wouldn't give it to children until you are very, very confident that this vaccine is safe and effective, because children don't die from this virus. So you
Yeah.
better make sure that you have a safe product before you put it into children.
You talked about antibodies and immunity, and we're correlating presence of antibodies and immunity essentially give protection. What are your thoughts on some of these antibody testing and a discussion that we can give out certificates of immunity based on whether or not they have antibodies? Is the science there right now to establish that?
No, you need to. You don't really know what level of immunity is protective. That's why you do prospective placebo-controlled efficacy trials, because then you may know that. I mean, we never knew that. With the rotavirus vaccine, even in 70,000 people, we still never had an immunological correlative protection, but we knew the vaccine was safe and effective, and now the vaccine is in millions of people, and we know it's safe and effective. But you often don't know that. So I think that to say, here's a person who has a certain level of immunity, therefore I know that they're going to be
Yeah.
protected. We're not there yet. I think I think we could get there.
How can we get there?
So by doing the kinds of trials where you
Okay.
see, okay, here's someone who's gotten the vaccine, they're protected, and we know this, they have this immune response. Here's someone who got the vaccine but didn't have that immune response and wasn't protective. So here's the immune response you need.
But how do we know how do we truly know they're protected? Do we retest over periods of time? What's the analysis there?
I think you know it in efficacy trials. I mean, by doing large prospective placebo-controlled safety efficacy trials, I think you can know what level of immunity, because you're looking at that. You're looking
Yeah.
at their immune response before they get the vaccine. After they get the vaccine, you'll see who's protected and who's not. Then you can hopefully have an immunological correlative protection.
Everyone's talking about reopening society and how we do that. And you had a very interesting editorial in the New York Times where you talk about crowding and the coronavirus, the key to reopening. And essentially you talk a lot about population density, and that's how we need to think about reopening. Can you walk us through that analysis?
Sure, that's the one thing that came through again and again. It is the degree to which the virus is being transmitted from one person to another. How common is it in that population? But if it's a population that's fairly spread out, you're at lesser risk in reopening, whereas a population that's very close together, as seen in large cities, you're at greater risk. Or a population that's close together in a meatpacking plant in South Dakota, you're at greater risk. So I think David Rubin, who really is the head of the policy lab at Children's Hospital Philadelphia, put this together, and I think he's made an important contribution.
And a key component to that is testing, correct? We have to do much more testing, much more frequent testing to sort some of these issues out.
Yeah, I mean, that certainly would help. I think in the final analysis, we're going to find that we're not going to be doing that testing because we've been talking about getting testing now for a long time and it hasn't happened. So I'm getting pessimistic about whether we'll ever have it. So I think what you need—
Rockefeller Foundation says we can do 30 million a week by June if we invest $100 billion.
I'll look forward to that. But in the meantime, I don't think it's going to happen. So, I mean, I think you have a sense of what the case rate is in a particular setting and how dense that setting is. And I think we're just going to see. You now have certain states like Georgia or certain parts of Florida or Texas or South Carolina that are opening things up, and I think we're going to learn a lot in this. So we're seeing New York open things up much more in the Germany model. I mean, New York has kind of acquired the Angela Merkel Germany model of let's do a lot of testing, both serological testing and who's shedding virus, and then knowing that we'll bring people out into the community, which I think is what New York State is doing. So I give Andrew Cuomo credit for that. But otherwise, I think we're just going to learn as we go, which sadly is also true.
Sweden announced this week that they think they have 30-40% herd immunity already, and that by June it may be as high as 50%. Is that realistic when we don't have a vaccine, we don't have effective treatments, we're behind on testing? How do we really know that?
I think Sweden made a mistake. I mean, they certainly have the largest number of deaths per million people in the world by choosing to do what they did, which was to take a laissez-faire attitude. Their bet, their gamble, if you will, is that they'll have enough population immunity from natural infection that they will then stop the spread of the virus. I don't think that's going to happen. I mean, remember, of the 4 human coronaviruses that circulate every year in the United States, they circulate every year in the United States. I think the only way we're going to get true population immunity, as Bill Gates says, is with the universal vaccine. I think that's right.
And then finally, you've been mentioning this is an RNA virus. Most of the time, they're not that complicated. But there's been discussion lately about reinfection. Do you think reinfection with coronavirus exists, or do you think that's a failure of accuracy of testing?
No, I think it may exist. I do think, though, that natural infection probably protects you against moderate to severe disease associated with reinfection, which is true a lot of these viruses, respiratory or intestinal viruses that have relatively short incubation periods where viremia is not part of pathogenesis. Usually you get protection that's short-lived and incomplete, but it is protection. I mean, a natural rotavirus infection will protect you against moderate to severe disease associated with reinfection. The same thing is true with the vaccine. I think you're not going to have a vaccine that does better than natural infection. That's typically true. It's the rare vaccine that does better than natural infection.
Well, Dr. Offit, I want to thank you for taking the time today to share your insights.
My pleasure. Thank you.
I want to thank you for watching Coronavirus in Context. I'm Dr. John Whyte.